Awesome. All right, so our first speaker is from Cal Poly Humboldt. It's Olivia, I'm sorry if I say your last name incorrectly, Boberitz, talking about the presence, distribution, and movement of green sturgeon in Humboldt Bay and the Mad River, California. All right, and I will kick it off to Olivia.
Okay, let me can you guys see just my presentation?
Yes, and also
Not
I'll give you a 5-minute warning when you're at the 10-minute mark, okay?
Okay, great. All right. Well, thank you everyone for joining. My name is Olivia Bobritz. I am a current master's student at Cal Poly Humboldt in the Department of Fisheries Biology. And today I'm going to talk about the presence, distribution, and movement of green sturgeon in Humboldt Bay in the Mad River, California. So, a little bit of background about my project. I'm part of a larger group called the North Coast Tribal Fisheries Collaborative. So, this is a working group of tribal, academic, state, and federal partners, and we have the shared goal of improving climate resiliency of culturally and economically important tribal fisheries in far northern California. So I'm one of 5 graduate students. I specifically am working with the Blue Lake Rancheria, and one of my main overarching goals is to increase their capacity for fisheries research by helping to fill in some data gaps and create new methodologies to monitor species of interest like the green sturgeon. So green sturgeon, there's a lot to say about this dinosaur fish, but I'm just going to highlight a few things for you guys today. So there are 2 genetically distinct population segments that range from the coastal waters of Mexico to the Bering Sea. There's a northern DPS in blue on this map, and they are characterized by fish that spawn in the Rogue River, Oregon, and the Klamath and Eel River, California, and they are considered of conservation concern. Then we have the southern DPS in orange, and these are characterized by fish that spawn solely in the Sacramento River and 2 tributaries, the Yuba and the Feather, in California, and they are listed as threatened under the Endangered Species Act. So we know that both of these populations like to hang out and aggregate in estuaries of non-natal rivers, so rivers they were not born in, as well as large West Coast bays in the summer to early fall months. We know this as one of the main methodologies that has been used to monitor sturgeon in the past decade has been acoustic telemetry. So there's a lot of fish from both populations out there with acoustic transmitter tags in them, and I'm going to talk about that just a little bit more soon. So I want to zoom in to this red star here. This is my study area. This is Humboldt Bay with the Eel River to the south and the Mad River to the north. So Humboldt Bay is the second largest embayment in the state of California. It also provides some of the most productive and dynamic ecosystems in the state. And this includes intertidal mudflats, eelgrass beds, sand dunes, riparian and coastal marsh, and forest. So the entirety of Humboldt Bay is listed as critical habitat for green sturgeon, but the eel and the Mad River are not. And Humboldt Bay also has several areas designated to protect wildlife, and I've highlighted one in that bottom left corner of South Bay, which is the South Humboldt Bay Marine Recreational Management Area. So I'm going to zoom in again to North Humboldt Bay and point out 2 sites of interest. So this red star is Sand Island. This is a small man-made island in the northernmost part of this main central channel in North Humboldt Bay. And this is where a previous telemetry study done about 15, 20 years ago and some word of mouth have shared some documentation of green sturgeon occurring. We also have this yellow star. This is the site of a proposed wind power terminal. So this figure on the left, that is what this construction, some of it might look like on the peninsula itself and into the water. It's important to think about that this channel is the only way fish can enter or exit North Bay. I've also included this figure on the right, which is from a Humboldt Bay shellfish aquaculture permit expansion proposal. This was approved in 2017, so all these solid white boxes you are seeing are existing aquaculture oyster beds. If you didn't know, Humboldt Bay is the leading producer of aquaculture oysters in California, and we produce over 70%. So what you are seeing in these 2 images is a lot of artificial infrastructure in and proposed to be in the bay, bays which are already common sites of intense human activities. So it's really important to look at this, knowing that artificial infrastructure does have the potential to interfere with ecological processes and potentially displace fish habitat. So it's important for us to understand how green sturgeon are using this area so we can understand how susceptible they might be to anthropogenic impacts. So given all this, I have a couple of research objectives. One, I would like to document the presence, distribution, and timing of green sturgeon in Humboldt Bay and the Mad River in that likely summer holding period. I would also like to determine the extent to which they're moving into the bay. So how far and which reaches are they using? And I'd also like to identify some potential aggregation sites. I'm looking to establish baseline data specifically at that offshore wind power terminal site. And I'd like to look more into individual movement patterns and how they relate with some environmental variables. So to do this, I have a suite of techniques. I have acoustic telemetry, drone and visual surveys, citizen science, and pop-up satellite archival tags. So first, I'm going to talk about acoustic telemetry. And in 2024, I deployed 18 acoustic receivers, 17 in Humboldt Bay, one in the Mad River, from August to December. And these were to detect fish that were previously tagged. So I did not tag any fish, but like I mentioned, there's a lot of fish out there with these acoustic tags. And these tags are the same frequency as my receivers, so they can communicate. And when a tag is near, it'll ping my receiver. And these tags, they are 69 kilohertz tags. They have a battery life of around a decade. So any fish tagged in the last decade has the potential to be picked up on these receivers. So the image here, all the red dots are the receiver locations. The two white dots, unfortunately, I lost these receivers. That's just part of the game. And I don't have any data to report on. But the breakdown is actually quite simple of what you're looking at. So South Bay, there are two main channels. So you'll see receivers in the bottom and the top of these channels, where the bottom corresponds to that MPA I mentioned and Hooked and Slough. You also see receivers in Elk River Slough, Freshwater Slough, and Mad River Slough. So this is looking into that objective of how far and what reaches of the bay are they coming into. And then I attempted to put receivers at any entry channel or exit channel to North Bay. And I really loaded up that main channel leading to that Sand Island site, as I was expecting green sturgeon there. And lo and behold, these receivers left on the map are the only receivers I got any detections on at all, so entirely North Humboldt Bay. And to break this down a little bit, so I had about 140,000 total detections. These were produced by 12 individual fish. So the map in the upper right, all the triangles are the fish, where the green is the data first detection at the receiver they were first detected at. So I deployed these receivers on August 20th. All these fish were first detected on August 20th, which tells me they were already there at that Sand Island spot. The red is the date they were and the location they were last detected, and this orange one, this fish was detected first and last specifically at this location. So I had fish leave as early as September 12th and as late as November 19th. The rest were pretty sporadic in between those dates. And if you look at the bottom image, this is a breakdown of those raw detections through this period, where there's a peak in September that gradually declines throughout October and then even more so into November. So you might be wondering, who are all these fish? Well, the 11 I showed are all green sturgeon. The 12th is a bonus. This was a broad-nosed seven-gill shark tagged by Oregon State University in the Puget Sound. And this is a bonus of conducting research like this, providing data to some collaborators and learning more about Humboldt Bay. And quite frankly, they're friends with sturgeon, so that's very interesting. And so here's just the sturgeon. This graphic on the left is showing you their movement between these receivers, where the blue arrows are going to correspond with northern DPS fish, so fish tagged in Grays Harbor or Willapa Bay, Washington. The red, orange, and yellow arrows correspond with fish tagged either in the Sacramento River or the Feather River, California. The one in black, I have not yet been able to identify the DPS of origin or the tagging location. But still, in this movement pattern, you can see they really are bouncing around that main channel, spending most of their time and kind of always returning to that Sand Island spot throughout this period. We can also learn a bit more about those raw detection counts. So you'll see that Sand Island spot to the north has the most, around 50,000 detections, that gradually decreases down that main channel. You also see the lefternmost channel, the Mad River Slough channel, had 8 fish visit there. No fish stay here for more than a day or two, so it's likely they were just passing by. But looking at this proposed some questions for me, like how are we jumping from 10 to 11 fish in these two locations? Where's that fish going and coming from? This is where I want to bring back those 2 lost receivers. Unfortunately, they were in kind of critical positions, spots very likely for fish to move in or out of the bay at. And also, at a high tide, there's no reason fish have to stay in these channels. They can absolutely transverse horizontally across the bay, which was this is all a great learning curve as I get ready for my 2025 field season. So on this map now, there are green triangles, and these are the receiver additions I'm going to put out much earlier this year, starting in May of 2025. You'll see I'm doubling up the Mad River. I'm incorporating more in the left and right channels of northern Humboldt Bay. I've turned those 2 lost receivers green, as I don't plan to lose them, and I do aim to deploy them differently to mitigate some receiver loss here. And then South Bay, instead of focusing on those two channels, making a gate just to be sure I'm capturing if anything is coming in or out. And I also will be incorporating the Eel River this year. You'll also notice a white diamond, if you can see it there in the center. This is a receiver that is currently out there. I deployed in January, specifically at that wind power terminal site. This will stay out there through the year, but is also acting as my, are there any green sturgeon moving in or out during the winter when we don't think they're going to be here? So in addition, in 2024, I did some visual surveys and some public outreach. I did not do any drone surveys in 2024 as permitting was getting set up, but those are set to commence in May of 2025 as well. So the visual surveys and the locations in yellow, I would bi-weekly go out during the same time my receivers were out, anchor the boat for 15 minutes, and look for what you're seeing on the left, which is flying green sturgeon, which is very common in North Humboldt Bay. And I would count how many I saw. Some days I saw up to 40 in this 15-minute period. Not individual fish, but jumping activities. I can't identify them to the individual. But I would also record some environmental variables like dissolved oxygen, water temperature, salinity, tidal stage, and time of day. Also during the same period, I was distributing flyers with a QR code linked to an iNaturalist project, and also with an email and some project information. And these were distributed on social media. I talked to fishermen. Matikit is a tour guide boat in Humboldt Bay. I also made some stickers to accompany this QR code, all with the goal of trying to engage the public into green sturgeon research and see if they would submit some observations of their own. So this map here, these are these two things together, where the yellow diamonds are my visual surveys, the only locations I saw green sturgeon, and the blue are all the public observation locations that came in. So these observations came in a lot of different formats, mostly jumping sturgeon, but we also had people see them under the water, sometimes with sonar. We had some incidental hookings in the halibut fishery. And these were all methods I did not use, so it did absolutely enhance my data set, give me more reach to determine observations. You'll see the public saw them in many locations I didn't see them, both the Mad and the Eel River, the Hooked and Slough, the Mad River Slough, and other locations in the North Bay. And this year is going to break down Humboldt Bay 2024 observations, except for the two rivers, the amount of sturgeon that we're seeing through the time, where the public observations are in blue and mine are in yellow. So you'll see the public saw green sturgeon in North Humboldt Bay as early as May. There was a peak in the summer because of the halibut fishery, perhaps because of tourism, or just because the weather is better to be out in the bay. When I was out there in the fall, it got pretty nasty. And this really highlights the importance of incorporating the community and doing some public outreach. Public observations nearly doubled my data set, and they also informed the reason I'm doing some things in 2025. So the public saw sturgeon in the Eel River, so I'm putting receivers in the Eel River. They also said sturgeon were in the bay as early as May, so I'm going to start in May. And some honorary mentions. I had some emails come in of sturgeon that were seen as early as the 1980s and the 1990s, which is just really fun to establish this network in the community. And lastly, we have pop-up satellite archival tags. This is last as I did not do any tagging in 2024, also as permitting was getting set up, but this is set to commence again in May of 2025, where I'll begin hook and line angling, likely in that Sand Island hotspot, to capture green sturgeon and tag them with pop-up satellite archival tags. So these tags communicate with satellites, right? And they're going to provide me a detailed track of location, time, temperature, depth, and acceleration. And they can stay on to up to a year before they pop up and float to the surface. And in the meantime, to determine how long and how well they're going to stay on, I've been practice tagging white sturgeon at the Cal Poly Humboldt Fish Hatchery. Also, I want to know any fish that is brought up will have a fin clip taken for genetic analysis to determine that population segment of origin. So I've thrown a lot of information out there really quickly. So I've brought back those main objectives to kind of highlight a few things we learned. So the presence, distribution, and timing of green sturgeon. My acoustic telemetry and visual surveys saw them aggregating in North Bay from August to November, but the public told me they were there as early as May. We also saw both northern and southern individuals. The extent to which they're moving into the bay, the public told me they are coming into some of those sloughs. They are going into those extremities. And that wind power terminal site, so any fish that wants to get to that aggregation spot in North Bay is going to have to pass this location. And that receiver, I'm hoping in 2025 will also tell us, hey, are they going to hang out and aggregate at that spot at all, or are they just moving past? And then 2025, there's a lot of future analysis I'm excited about. Most of this will be focused on seeing if I can find any statistically relevant trends with those environmental variables with all of this data. I will also be looking more into movement with that larger receiver array and those satellite tags. And drone surveys will begin, which will be really interesting to compare how well did the drones observe and count green sturgeon versus my eyeballs. And can that enhance more observations as well? So thank you so much. There's a lot of collaborators on here, but I'm like literally at the 15-minute mark, I think. So I've included my email and the Green Surgeon email in the corner, as well as some QR codes of the relevant things I've mentioned throughout this presentation. So thank you so much.
Awesome. That was great. And you still had like 7 seconds to spare. I'm sorry if I didn't know how to like best like let you know about time. So hopefully that was okay.
That was perfect. Thank you. That was great.
Okay. I'm like used to doing it in person, so this is my first time trying to do it virtually. But that was great. We still have some time for questions. I know I gave us a little bit of late start because of the announcements. So we're just running a little bit behind, but if anybody has any questions for Olivia, we still have a couple minutes for that if anybody would like to ask them. Great study. Lots of cool work you have coming up.
Thank you.
And lots of cool work you've been doing. I love seeing sturgeon breach, by the way.
It's one of the best things, I
It's
think.
so
Yeah, for
cool.
sure.
I've seen it once only, and it was amazing.
Yeah, when we go out there in the like September, August time, it's just like it's so hard to only do the surveys for 15 minutes because it'll be all day just flying around you.
That's super cool.
Mhm.
Any questions for Olivia? Tyler, you got one.
Um, yeah, great presentation. Very informative. That's really cool, especially the use of the different methods. So my question is, how much communication do you have with the folks that are tagging the green sturgeon? So like, do you know if they wander into Humboldt Bay and get detected on your receivers, you know where they came from?
See, some of them, yeah. So all of the data, like the people that tag these fish, they're collaborators to me. They are providing me the information on the fish. And I'm in the process of learning more every day about these fish. But yeah, specifically the northern DPS fish, Laura and Matt at Washington Fish and Game, they have amazing arrays up in Washington and can actually show me exactly the path that my fish in Humboldt Bay took across the entire West Coast. And same within the Sacramento River, there's a great receiver array there. So yeah, absolutely. I can see what they're up to for sure. And it's fascinating. And there definitely needs to be more receiver arrays in Northern California. And hopefully my receiver array will be continue to be maintained long after I'm gone by the Blue Lake Rancheria.
Awesome. Thank you.
Mhm.
Super cool. What a fantastic history you're able to get. All right. Well, with that, we will go ahead and switch to our next talk. Great job, Olivia. Fantastic. So our next student is Paige Gardner from UC Santa Cruz, who will be speaking to us about rapid evolution in the face of a changing climate. Can salmonids keep up with the rising temperatures? This one I'm particularly keen on.
Hi everyone. Sorry, I'm just going to go ahead and share my screen here. Let's see.
And I'm going
Share.
to do the timing the same way that I did for Olivia's, if that was that okay for you if I just
Absolutely.
okay.
Can you see my presenter view or slides?
I can see presenter view.
Okay. Any tips on how to fix that one?
Usually it's like
Let's see.
sharing a different screen is usually what I do is I have presenter view on one screen and then the slides on the other.
Okay, totally. Let's see here. All right, so just go ahead and
That's my
share.
solution. I don't know if anybody else has a better solution. And I haven't started
the-
the timer. I'll start the timer when you start speaking.
Okay, perfect. Thank you so much. Apologies for that. Um, let's go ahead and share that maybe, and then here, presenter view. How about now?
That's still going to be your presenter view. I'm sorry.
Sure, totally, totally. Sorry, everyone. Um, All right. How about now? Good to
There we
go?
go. Perfect.
Awesome. Thank you. Thank you for bearing with me on
Oh
that.
yeah, no
All
problem.
right. Okay, great. So let's go ahead and get started. So thank you so much for having me today. My name is Paige Gardner, and I'm a second-year PhD student with Eric Kalkovacs at UC Santa Cruz. I'm also the president of the Santa Cruz Monterey Bay Area Student Subunit of the American Fisheries Society. And today I'm going to be talking to you about the encroaching threat of rising stream temperatures on steelhead salmon in a warming climate. And the main question in the work of my PhD dissertation is if steelhead can keep up with the rate of climate warming. See, there we go. All right. So in addition to being ecologically, culturally, and economically important, steelhead have fascinating life histories. So Oncorhynchus micus, their species name, have two major life history forms. The resident rainbow trout that you see there in the bottom right picture that spend their entire life in fresh waters, and the anadromous steelhead in the top picture that starts its life in freshwater before migrating out to the ocean for several years to grow, and then migrating back up to its natal streams to spawn. Unlike salmon, steelhead can make multiple trips throughout their lives to the ocean, ultimately traveling thousands of miles. However, humans have placed immense pressure on steelhead and other salmon species. I like to sum these pressures up in this 5H's schematic. That would be hydropower, habitat loss and degradation, hatcheries, harvest, and then what I'll be focusing on in my talk today is heat. And then, of course, these 5H's are all linked by one intrinsic H, humans. So this map here shows the current and historical distribution of steelhead. The green is their current distribution, and the blue is their ocean distribution, but the orange and the yellow are where they have been locally extirpated or are high risk of local extirpation in the future. And as the most southernly distributed salmonid, we see a lot of their range reduction has been at their southern edge, and it's at least partially due to heat. And the threats and impacts of heat on steelhead is a rapidly growing area of study and has gained mounting media attention and research appeal as the effects of climate change become more and more realized. So in fact, a recent study found that 90% of Western United States steelhead trout populations are predicted to become thermally stressed with climate change. This is really concerning and is really urges more research to understand how steelhead populations will respond to climate-induced warming. But briefly, I do want to discuss how heat negatively impacts fish. So steelhead, like many other aquatic species, are ectothermic, meaning they rely on the temperature of their environment to survive. So they rely on the presence of cold water to maintain healthy physiological processes, such as escaping predators, foraging, metabolism, reproduction, and survival. I included this figure to kind of depict the effects of heat on their ability to complete those physiological functions. So as you can see, as temperature increases, moving left to right on the x-axis, the fish's ability to complete that physiological function depicted by the red line steadily decreases until eventually it's gotten so hot that they completely fail and are no longer able to complete that physiological function. So if you're for migrating steelheads, migrating long distances, having extra heat can inhibit these processes and be the difference between life and death. To avoid extinction, as climate change continues to warm rivers and streams and place pressure on their physiological capabilities, fish populations need to find a way to survive and adapt. So thermal tolerance is a really complicated trait, and that many things can contribute to variation in thermal tolerance both within and between steelhead populations. Thankfully for salmon, there are several different pathways for population persistence that may offer refuge from environmental warming. Here, thermoregulation is the ability of an individual to maintain its body temperature, and thermal tolerance is the temperature range at which individuals can maintain physiological processes needed for life. And thermal tolerance can be mediated by complex cellular processes, but it can also be a function of behavior and individual level heat responses. And today we're going to be looking at it broadly, looking at three different pathways of persistence, being adaptation, plasticity, and behavioral thermoregulation. However, while behavioral thermoregulation is really interesting and an extremely important mechanism for survival, I am not going to be discussing it in my talk today. Instead, I'm going to be focusing on adaptive potential and plasticity of thermal tolerance in steelhead populations in California specifically. So firstly, to address plasticity, I want to understand how the thermal tolerance or the upper edges of a fish's thermal limit can be impacted by its environmental conditions. Next, I want to understand the role of adaptation and thermal tolerance. Are populations from warmer streams better able to tolerate high temperatures than those from streams with cooler temperatures, indicating local adaptation? And the second part of that question is, are there specific genomic regions that are associated with differences in thermal tolerances, both within and between populations? Now, this particular adaptation portion of my study is an ongoing area of my research. So today I'll be talking about what we've learned so far, our hypotheses, and the next steps in this section. And then lastly, I want to understand how we can use what we learned from our both physiological and genetic studies to inform conservation decisions. And these three broad questions are all contributing to one major question is, can steelhead keep up with the rate of climate change? Let's get into how we designed the study. So to try to answer this question, we selected 3 different sites in Central California. All these sites are ESA listed as threatened, and they're all in the Bay Area. So first we went to Big Creek down in Big Sur. The next we went to Carmel in the Monterey area, and then lastly we went to Scott Creek up in the just north of Santa Cruz. We selected these sites based on their very different thermal regimes. So Big Sur, our southernmost distributed sites, had consistently very cool temperatures. And this plot here is showing the historical average stream temperatures, including average daily averages, minimums, and maximums over the time series available. So Big Sur is our coolest. And also I want to point you especially to that middle peak in the summer months, because that's when we did our studies. And the Carmel River had extremely high average temperatures in the summer, but not much variation throughout the day. And then Scott Creek had very warm temperatures and extreme daily fluctuations in temperatures as well. So at each site, we sampled 100 individuals. And this is just to show the distribution of the size of the individuals that we sampled. We were targeting juveniles specifically. So we were looking at ages 0 to 1 plus, ranging in general from like 50 to 200 millimeters in length. Okay, so this study was designed to take place entirely in the field. So this allows us to test fish as close to their natural environment as possible. And there are 3 key steps to this protocol. First, we hold fish in temperature-controlled tank streamside to induce a thermal response to an acute warming event. So we divide this group of 100 into 4 groups of 25. And then we hold each of these different temperature treatments. So first we had our control ambient. Our next treatment was held at 20 degrees Celsius, and then 23 and 25 degrees Celsius. And each treatment group was held for 20 hours at this temperature before beginning experiments. And this is what this looked like in practice, where the fish were held in that top tank and stream water was heated in the tank below it on the left. So next, we experimentally measured thermal tolerance using critical thermal maximum, or CT max. And to do this, we placed a subset of the fish into a container, and then we heated the water in that container at a rate of 0.3 degrees Celsius per minute. And the trial was over for each individual fish once they lost equilibrium. So that's the temperature at which they were no longer able to stay upright in the water column. And this is what this looks like here, where the fish are placed in that open container, and that's where the water is heated. And then lastly, those fish are allowed to recover. All these experiments are non-lethal. They're allowed to recover, and then they're taken for length and mass measurements, and then we take a thin clip for genetic analysis. Okay, so firstly, to address plasticity, I want to understand how exposure to an acute warming event changes thermal tolerance within a steelhead population. So we would hypothesize that increasing acclimation temperatures will induce higher thermal tolerance, causing higher CT max values. So here on the x-axis is the temperature treatment at which each of these groups held for 20 hours leading up to the experiment, and the y-axis is their critical thermal maximum. So we would expect to see these values increase as we move right across the graph. And that's exactly what we saw. So just to orient ourselves more to this graph, each little dot is an individual fish. The large dot is the average critical thermal maximum in each treatment. Pink is Big Sur, Carmel in green, and Big Sur in blue. So we see that all 3 populations had this plastic response. So then thermal tolerance is impacted by environmental temperatures, and its environment can impact its thermal tolerance and has some degree of plasticity. All right, so next, to understand the role of adaptation, we want to understand how populations from warmer streams tolerate high temperatures compared to those from cooler streams. Are there specific genomic regions associated with thermal tolerance? And again, this is an ongoing area of study, so I'll be presenting what we've learned so far from our first field season and the next steps and hypotheses. So we hypothesized that steelhead populations from warm streams can tolerate higher temperatures than those from cool streams. We might expect some sort of like thermal performance curve that looks something like this here, where individuals from cold source environments need much cooler temperatures to maintain high performance, while individuals from hotter environments can maintain high performance at higher temperatures. So looking back at that same figure we looked at before, we can begin to see some variation within treatments around the means. So preliminary data analysis shows some critical differences between the mean critical thermal maximum of each treatment between populations, and that's indicated here by the asterisk. So you see that Scott Creek has, on average, in treatments 2 and 3, higher critical thermal maximum than the other two locations. And just to like revisit our temperature sites, we said that Scott Creek is warm and variable, Carmel is warm but constantly warm, and Big Sur is cool. However, the differences in the means within these treatments is still like fairly minimal. There's still less than a 0.5 degrees Celsius in magnitude difference between site mean variation. So while the differences in the mean between populations is certainly interesting, what is even more interesting is if we zoom in on one of these treatments to look at the variation around the mean and look at within population responses to heat. So looking at treatment 3 here, we see that in Scott Creek, almost all the individuals had the same thermal response to a heat event. They all had almost the same critical thermal maximum. There's not much distribution around the mean. However, when we look at warm, sorry, that's incorrect on the slide, but warm and constantly warm site in Carmel River, we see that there's a huge distribution of critical thermal maximum values around the mean, ranging from 30 to 32.5 degrees Celsius. So biologically, we can say that all individuals tested in Scott Creek had nearly the same physiological response to warming. And in a warm environment with extreme daily fluctuations in temperature, it may be beneficial to have high plastic thermal tolerant traits. However, in Carmel, not all individuals have the same temperature response. So the difference is, this means that there might be some individuals that are way better at tolerating high temperatures than others in the population. So this has important implications for the genetic portion of our study. And again, this is like our plans for the future. This is an ongoing work that will hopefully start in the next year with our collaborators at NOAA. So what we can do is we can correlate variation in critical thermal maximum with variation in the genome. We'll select individuals on either end of the extremes, having really high thermal tolerance and those having very low thermal tolerance. And then we can correlate that using genome sequencing with genetic outliers. And we would generate some sort of plot like you see there on the bottom, where each of those dots indicates potential genetic regions associated with differences in thermal tolerances between individuals. And this is really important when we want to understand the rapid evolution of thermal tolerant traits. All right, so keeping on moving forward in the interest of time. So inter and intra-population variability in thermal tolerance potentially suggests local adaptation to warming. And this is very promising when we're thinking about rapid evolutionary potential. So then how can we use this information to inform conservation decisions? Ultimately, we want to ensure that the results of the study can help inform conservation outcomes that this portion of the study will come in later steps. And all this research is supported through NOAA. And as a NOAA affiliate and coastal science and policy student, I'm in a unique position to interface with managers and policymakers. And the goal of this study is to provide tools that allow managers to target their restoration efforts and come up with new conservation methods to help preserve steelhead in the face of climate warming. So for example, we could target habitat restoration on streams that are at high risk, or the streams that have populations with high risk of extirpation due to warming. So we could create preserved thermal refuges doing things such as building log dams like you see in the picture there on the left, which channelize flow to maintain high flow throughout the year, and also creating cold thermal refuges that are very important for survival. We could also prioritize keeping water in rivers during the hottest times of the year by monitoring how much flow is taken for like irrigation and other purposes. And then the most challenging conservation strategy is genetic rescue. So we could use this research to identify genetically vulnerable populations that don't have the genetic variation needed to adapt to the next century of warming. We can also identify genetically valuable populations that convey thermal strength. And as depicted in this figure here, we could introduce individuals from a different source, thermally strong population, to one that maybe is in danger or already going extinct due to high heat. And then here we see just like some southern steelhead might be a good candidate for either genetic rescue, and that they may need improved alleles to convey improved fitness in the face of warming. Or maybe the southern steelhead populations have really high thermal tolerance and alleles that convey rapid adaptation in the face of warming. All right, so I think I have a little summary slide, but I'm going to keep moving to my acknowledgements in the interest of time here. So I want to thank everyone and all my collaborators who have helped with this project, and all of my collaborators at NOAA, the Fisheries Collaborative Program, and my wonderful volunteers. And with that, I will stop and take questions.
Awesome. Great job, Paige. We have time for some questions. So if anybody has any questions for Paige, right now is perfect. I love the fishies that float up when Kim does it. Sorry, it just entertains me every time. All right. That's a lot of cool work that you're doing there.
Oh, thank you.
And a lot of really pretty areas. This is like a gorgeous picture. So
Oh, we're super lucky.
yeah, very
Absolutely.
fortunate to work in some cool cool spots for sure.
It definitely helps.
Yeah, for definitely. Anybody have any questions for Paige? Must have answered everybody's questions during your talk.
I'll say that this is an ongoing, like we're really excited for the 2025 field season that we can expand our sample size and sample sites. And yeah, I'm excited to expand the genomics portion of this project.
Cool. Well, hopefully you'll come back next year and give us an update then.
Yeah, that's the goal.
Perfect.
Absolutely. You'll all be present throughout my dissertation. So
Can't wait. All right. Well, if nobody has any, oh, wait, we got one from Matt. Never mind. Spoke too soon.
Uh, you kind of led into my question. I might have missed it, but can you talk about what your new sites are going to be?
yeah, absolutely. So this has been a high area of debate with my collaborators. We want to make sure as we're selecting new sites that it sets us up for success, not just in the physiological aspect and understanding differences in physiological responses to heat, but we want to make sure we're also like setting ourselves up for success in the genetics. This is a very like challenging portion of the study, and so selecting sites within like hopefully within like similar genetic regions. So there's like south of San Francisco Bay will set us up for like the most success. And so we're looking at sites in um, like the San Lorenzo River and some other sites potentially as well, like further north in the Russian River. But the goal is overall to like expand our understanding of how different populations are responding and then how that is reflected in their genetic signatures as well. So not a great answer, but this is definitely like an open area of discussion that we're doing our best to like make informed decisions.
Yeah, that was a great answer. It's nice to know there's so much thought going into that. That's great. Super excited to hear more.
Yeah, thank you for your question.
Awesome. Well, in that case, great talk, Paige. And we are going to move next to our next student, who Rachel Hine from Cal Poly Humboldt, who's going to be speaking on the spatial, temporal, and ecological influences on the occupancy of juvenile Chinook salmon in the Eel River estuary. So Rachel, I will hand it over to you, and then just be on the lookout for my little flashing numbers.
Sounds great. Let me just share my screen here. Okay, are you seeing the presenter notes at all?
Not seeing the presenter notes, or seeing it's like a smaller
Okay,
version, like a Google slide.
let me see if I can troubleshoot that. One second.
Yeah, no worries.
Okay, is that doing it?
Taking a second to
Still
show.
small.
Well, it's taking a second to show. Oh, there we
Oh.
go. Yeah, and it's still like well, it looks like it's a tab in one of your um there we go, it's bigger.
Okay, is that full screen?
I think so. It's like most of the screen.
Okay.
I would say like 80%.
80%. I'm sorry, I don't
Right,
share.
which I think it's probably good enough, right, guys? I don't know. I can see enough.
Yeah, Google Slides always does this where you see the header of the website, but we're seeing the full screen,
Yeah,
which
we're
is
seeing full screen.
Okay.
Yeah. No, you're great. Sorry.
All right. And can you still see full screen here?
That makes it like a little bit smaller.
Oh man.
The other
Okay.
way you had it was bigger. I mean, it still works.
Sorry, guys. Is it still small?
I think it's good.
Okay, we'll just move it that way.
Sorry.
Sorry about that. Thank
That's okay.
you for your patience. I am Rachel Hine. I am a master's student at Cal Poly Humboldt in the Fisheries Department. And I will be presenting today on my thesis research, which is the spatial, temporal, and ecological influence on the occupancy of juvenile Chinook salmon in the Eel River estuary. Oops. So just to start out, I'd like to give a quick outline. I will first talk about the background and rationale of this project, move on to methodology, and then the planned analyses, and share a bit of descriptive data from my first field season of sampling, and finally end on the next steps for this project. So before I begin, I'd like to give land acknowledgement to the Wiyot. So my study site is located on the unceded ancestral territory of the Wiyot peoples, which includes the Wiyot tribe, the Bear River Rancheria, and the Blue Lake Rancheria. And the ecosystems that surround the estuary are the result of the indigenous land stewardship that has shaped this area since time immemorial. Wiyot people continue to be an integral part of these landscapes. And this project in particular is partnered with Blue Lake Rancheria as part of the North Coast Tribal Fisheries Collaborative Group, which Olivia spoke on a little bit earlier. But we have the shared goal of improving climate resiliency to culturally and economically important species here on the north coast of California. And my study species, Chinook salmon, are of particular importance for sustenance, livelihood, and ceremonial purposes for Indigenous people, and is an important component to include when speaking about this work. So just keep that in mind as I move forward. Oops. And I just wanted to start by outlining the guiding questions that really guided this thesis work. And those are juvenile Chinook salmon utilizing the Eel River estuary? If so, how are they distributed on the landscape spatially and temporally? And what factors might be influencing those observed distributions? So just some key background points that are important to think about when answering these questions, that estuaries are well known as nursery grounds for a lot of juvenile fish, and this is no exception for juvenile Chinook salmon. They are an estuary-dependent species and have been well documented utilizing estuaries north of California, and they exhibit life history diversity that can be seen in the timing and size at freshwater immigration, ocean entry, and in their estuarine rearing strategies. And these strategies can buffer against unpredictable environmental conditions and distribute risk across their populations. For Chinook salmon, this can be seen in the age and timing of their downstream migrations. And movements and residency within the estuary can be dependent on estuary conditions, as well as individual size and growth. Residency periods in productive wetlands can provide adaptive strategy that increases the size of juveniles and ultimately their survival at ocean entry and into adulthood. And also estuaries can offer heightened foraging opportunities, a longer period to adapt to higher salinity levels for juvenile Chinook, and offered a buffer against upriver conditions. So a little bit about my species in particular. The Eel River Chinook salmon are part of the California Coastal Evolutionary Significant Unit, and they are currently federally listed as threatened under the Endangered Species Act. And this is because this species has endured a drastic population decline up to 95%. And this is largely attributed to the combined effects of logging, overfishing, habitat degradation, invasive species introductions, as well as water diversion projects. And in the estuary in particular, there's been a lot of sedimentation that has accumulated from the higher gradient reaches upstream and has really narrowed that tidal exchange. And this has further been intensified by leveeing and diking within the estuary for agricultural purposes. Previously, there has been a few reports on juvenile Chinook salmon within the estuary. Most recently, there was one conducted in 1995 by Kanata and Hassler, and previously one in 1977. So now I'd like to introduce you to the Eel River estuary. So the estuary is up near Fortuna in Northern California, if you know where that is. And it drains the Eel River watershed, which is the third largest watershed entirely in California. And the estuary itself is usually split into 5 different regions. So you have an upper estuary, a middle estuary, a lower estuary, which you can see in the map here. And then you have the northern slough channels and the southern slough channels. And the Eel River estuary has experienced a lot of changes since the last study that occurred in 1995. In particular, there has been 2 large-scale restoration projects that were completed within the last decade. And there's also 2 more that are planned to take place within the next few years. And also of note is that there has been a spread of invasive species, particularly the Sacramento pike minnow, which is native to other drainages in California, but was introduced into the Eel River in the 1970s and is hypothesized to further impair the Chinook salmon recovery as it preys, has been known to prey on salmonids within the Eel River. So the objectives of this study are then to identify seasonal and spatial patterns of juvenile Chinook salmon abundance and distribution in the different estuarine habitats, use size spectra analysis to explore potential ecological factors structuring communities, and then look if there's indicative spatial gradients in productivity, predation risk, habitat, and environmental characteristics that might be explaining these occupancy patterns. So the estuary itself is comprised of a mosaic of different habitats that can be classified into 3 categories. So here we have the primary habitat, which is generally the main stem Eel River. We can have secondary habitat, which are large sloughs that branch off the main stem. And then finally, we have tertiary habitat, which are little tidal wetland channels or blind end channels that branch off that secondary slough. This is a figure of all of my sampling locations within the Eel River. So you can see they are very wide reaching. This is a large area to sample. Generally, my sites are split up between primary, secondary, and tertiary in equal amounts. And just to give an idea of what some of these habitats look like, this, for example, would be a tertiary habitat. So this is sort of a blind end pond. And then we have some examples of larger secondary slough habitat. And finally, some main stem sites where you can see there's very wide channel in these spots. To jump into some of my sampling methods, I've broken this down by occupancy and size spectrum. So for the occupancy component, I take water quality measurements at each site, which is dissolved oxygen, temperature, salinity, and depth. And at each site, I complete 3 non-overlapping seine hauls that serve as replicates for the occupancy. And if we get Chinook salmon, we measure them and we weigh them. And the plan is to complete an occupancy analysis with the goal of determining occupancy and detection probabilities and how the potential covariates might be structuring that occupancy for juvenile Chinook salmon in the Eel River. And if you're familiar with occupancy, you'll recognize psi as the probability that a site is occupied within the estuary and P as a detection probability or the probability of capture in a seine haul given that they are present at that site. And the size spectrum component requires a little bit more in terms of sampling. So for the size spectra, we will measure up to 30 individuals per species per site, and then count total individuals per species in each of those seine hauls. We then will convert the length to weight by using species-specific length-weight conversions found on FishBase, and then create a data set that has the total biomass per haul, which is generated by sampling with replacement from those 30 individuals. So size spectrum theory is based upon the observation that in pelagic systems, you have a lot of smaller individuals in the system and less larger individuals, but that the total biomass remains relatively constant across size classes. So if we think about this, we can plot the log of abundance against log of body mass and create linear regressions based off this size spectrum. And we can extract different parameters from these regressions, such as the slope that can be served as a proxy for predation and the intercept, which can serve as a proxy for productivity within these habitats. So we might also imagine a scenario where you have less smaller individuals and more larger individuals. And if you were to create a linear regression from this, you would have a shallowing of that slope, which equates to a higher predation pressure. So we can see, or you can imagine that in the estuary, you might have these different linear regressions that are structuring different habitats based on the community that is present. And you can imagine how that might change given spatially or temporal distribution within the estuary. So for this study, we'll be using these linear regressions to infer the different properties of community structure and nursery function throughout the different habitats and compare those intercept and slope parameters. So my hypotheses and expected results then for each component is that the occupancy, the presence and abundance will differ based on sites and will change with growth and ontogeny. So I predict that there will be younger, smaller individuals in the off-channel wetland habitats earlier in the spring and winter, and larger individuals will come down later. For size spectrum, we will have higher predation risk in the main stem, as opposed to lower predation risk, but higher productivity in that off-channel habitat. So then I'd like to just present some results from our first season of sampling. So I did a compacted season of sampling pretty much through the summer of 2024, but in total we conducted 144 seine hauls, captured 12,689 individuals that represented 21 unique species. Of these, the top 18 are plotted here, and you can see our study species, juvenile Chinook salmon, were the lowest, but they still made it within the top 18. And the species caught the most often was top smelt, which included both adults and a lot of juveniles. And there seemed to be little overlap between the adults and juvenile habitat, which suggests that the species composition was variable between our sites and across habitats, indicating that there is spatial temporal shifts in fish community and nursery capacity in our different habitats surveyed. And for Chinook in particular, over the course of that season, we captured 25 individuals. And this figure here can show the sampling scheme that we did have. So the X's were not sampled, the dashes were sampled, but no Chinook were documented. And we also see that the majority of individuals were captured in June, and that we captured no individuals after July, suggesting the migration had ended. Our naive occupancy estimate was 8 out of 24 sites. So of the sampled sites, juvenile Chinook occurred in 8 of those. And we can break this down in a different way. So if we look at where these juvenile Chinook were mostly located, we can see the majority were in that primary main stem habitat. But if we look at this seasonally, it tends to show a different story. So the first graphic here on top is March and April, middle, May, June, bottom is July. And we can see there's a shift from those tertiary and secondary habitats to primary later in the season. And this really highlights the critical importance of having habitat heterogeneity and the connectivity of those habitats for juvenile Chinook salmon through their migration period. And this is also an interesting graphic that shows flow on the right side, CFS, and then the fork length of captured individuals. And just to show a few examples of individuals we did catch, you can see that the smaller individuals tended to show up earlier in the season, and there's a direct trend to larger individuals coming down later. And it is interesting to note that the smaller individuals showed up after a large flow event, suggesting that they may be carried into the estuary by large flow events early on in the springtime. And this shows some of that life history diversity that I spoke about earlier, and it'll be interesting to see if this plays out forward for this sampling season. So the next steps then are complete my primary field sampling season this year. I started in February and will continue sampling through September, and then conduct those data analyses with the data that I collect this year, so size spectra, Bayesian occupancy, and I would love to compare against previous reports and really tease out differences that have occurred since the last couple of reports that happened. And lastly, I just want to touch on the importance of this project. So like I mentioned, utilization is largely unknown. We have these previous reports, but they have not been looked at in over 30 years. And really, this project will help understand how habitats are essential to guide effective conservation restoration efforts for the California coastal Chinook salmon. And I hope that once complete, my findings will be used to evaluate the effectiveness of recent restoration efforts, guide these new restoration efforts that are coming down the pipeline, and identify important rearing and migration corridors for not only juvenile salmon, but other fish that utilize the estuary. And with that, I would like to thank my advisors and all of my collaborators and field help for this project. It's definitely a big undertaking, but I'm really excited for the next season to see what we get.
All right, fantastic talk, Rachel. Everyone's got such beautiful field sites. I'm very envious because I worked in a lot of rice fields in the Central Valley. I should have gone to more coastal areas with you guys to do some my salmon work. All right, does anybody have any questions for Rachel before we turn it over to our last speaker? Matt Young, got your hand up?
Okay, I have a question. So I want to confirm when you were looking at fish size, right? You're measuring the length, but inferring weight from a length weight regression.
Yes.
So, or thinner fish might
I think you might
influence.
have cut out for part of that question, Matt.
Regression is to look at
Yeah, I'm gonna just go ahead and speak for Rachel and that we can't understand you.
Sorry, Matt. My email's up here. Feel free to email me your
Or
question.
yeah, or put your question in the chat maybe.
That would work.
I heard something about thinner and regression. I think those are like the only words I caught. I'm gonna go ahead and skip to Kim for right now. Kim Luke.
It's my turn. I was wondering, so you were doing beach seines, correct?
Mhm.
Do you have like are you considering any other sampling methods to maybe get like especially in those like larger, wider areas? Or are you just assuming they're just in the nearshore habitat? I just was wondering if you were thinking of like deploying different, I know that would like mess up your occupancy models potentially if you're using different samples or like methods for sampling. But wasn't sure if you thought like the lower numbers might be due to the type of sampling or something else might be more efficient or effective.
Mhm. Yeah, that's a good question. I think it's very difficult to sample in the estuary anyways. There's a lot of different factors to play into, even for seining, like tides can change it. But we try to sample as best we can. We have different nets that we use for different habitats. So we use a pretty large 100-foot seine for those wide main stem habitats, and we get a pretty good portion. And then for my sites, I usually have, for at least the main stems, I have paired sites on either side. So we're capturing most of that distribution of the wide channel. But yeah, I think seining in this case is the easiest way to capture those individuals, and sort of constrained by my field. I guess I think the other way to do it would be by like trawling by boats, but I'm kind of constrained by using just kayaks. So I'm limited in the options that I can use for that.
Got it. Thank you.
Awesome. Thank you so much. So Matt's question was, can you talk, and this will be our last question. So can you talk about how measurements of actual biomass weighed fish rather than inferred estimated from regression would influence the size spectra analysis? Tidal wetlands can make fat, happy fish.
Yeah, they can. That's an interesting question. I think we are mainly looking at different length to weight regression. We're using Bayesian coefficients that are on fish base, so it accounts for the different body sizes for different fish. And that was one thing we were thinking about, maybe just using the length, but in the estuary, there's different body types for different fish. So that's why we are converting to biomass. But yeah, I think definitely you could have fatter fish, but it accounts for the best way to do that in the way that we have available, if that makes sense.
And there's also some really cool morphometrics work out there. Looking at some of like Eric Combs' work at UC Davis when he was there, of like taking pictures of the fish and like calculating and having like the different points and then actually estimating the weight
Mhm.
based on the morphometrics analysis. There's some cool, he published a cool paper on some of that.
Look into that.
Yeah. So that was a great talk. We'll go ahead and move on to our last speaker. Thank you, Rachel, and thank you everybody for sticking around. So our last speaker is Matthias Milton from CSU Monterey Bay Moss Landing Marine Labs, and he is going to be speaking on the biochemical indicators of upwelling stress in adult gopher rockfish in Central California. All right, I will let you
Awesome.
take it away, Matthias.
Awesome. Thank you. Let's see. I'm going to share my screen. Whoa, spoilers. Okay, there we go. Okay, hi everyone. Thank you for that introduction. So my name is Matthias Melton, and today I'll be talking about my research on biochemical indicators of upwelling stress in adult gopher rockfish in Central California based on our lab and field studies. So first, I want to define biochemical indicators and then talk about how we use them. So biochemical indicators are a set of biological processes used to test if an animal's condition or stress is tightly coupled with variation in the environment. So here we have a figure to the left by Alfonso et al. 2020, where a fish is reacting to heat stress, and the fish is going to produce heat shock proteins in response to that stress. Now, we don't need to know all the mechanisms, complex mechanisms within this fish. We just need to know that heat stress is coming in, the fish is reacting to that stress, heat shock proteins are produced. Now, if we can correlate the production of heat shock proteins to the amount of heat stress that the fish is undergoing, there's evidence to support that those heat shock proteins are biochemical indicators of a fish having a heat stress response. And so the variation in the environment that I'm looking for in this talk is upwelling. So seasonal upwelling happens in Monterey Bay during springtime, and it occurs when wind blows parallel to our coast, allowing for warm surface layer waters to blow offshore. And this allows for cool, CO2-rich, oxygen-poor water to rise up and take its place, ultimately creating ocean acidic and hypoxic conditions for nearshore organisms like the gopher rockfish. And climate change may increase the intensity and frequency of upwelling events in parts of the California current system. So this is very important to study the effects on gopher rockfish. So moving on to the bioindicators of this project, the first one, we have HIF-1 alpha, or hypoxia inducible factor 1 alpha, and studies have shown that this is a proxy for a hypoxia response within the fish. L-lactate, which is a proxy for metabolism, specifically anaerobic metabolism. Cortisol, which is a proxy for the overall stress of the fish. And then IGF-1, which is a proxy for growth within the rockfish. And so the big theme of this talk is, always thinking about, can we use these as bioindicators of upwelling stress in rockfish species specifically? So let's meet the gopher rockfish. They are a nearshore and benthic species. They like to hang out in the rocky reefs, in the crevices of kelp forests and such. But they also like to stay stagnant. They like to stay in the same area from when they were born. So they are potentially more susceptible to those upwelling flexes and therefore low oxygen and low pH stress. They're commonly caught, making them a great study species. So we can catch a bunch of them, bring them to the lab and test them. And then they also have a unique reproduction called viviparity. And so they have fertilization occurs internally, and then they give birth to live young. And so the main point, though, is that the reproductive timing overlaps with the seasonal upwelling we see in the Monterey Bay. So the reproduction is January to May, and the upwelling season is between March and August. So going to the overall question of this talk is, how does regulatory proteins in adult gopher rockfish differ across varying levels of acclimatized upwelling exposure and duration in the lab and field? So we have our indicators here, and for HIF1 alpha, L-lactate, and cortisol, we would expect increases in the concentrations of those proteins within the tissues of the rockfish in accordance with upwelling stress. And then IGF-1, we would expect the opposite. So if a rockfish is in upwelling stress, IGF-1 levels would go down, and we would see that within the proteins of the tissue post-mortem. So moving on to the methods, we had to go out and catch these fish in the Monterey Bay. So we went in the spring season during the upwelling season of 2023 and 2024, and we collected the rockfish off of Point Joe. So here's a map of roughly where we caught the fish. So for the laboratory study in 2023, we brought them back to the lab, acclimated them for about 2 weeks, and then we exposed them to different treatment levels for about 5 months. And we had 20 fish per treatment with 4 fish per 5 replicant tanks per treatment. And so we have ambient, moderate, high, and extreme. And these levels of pH and dissolved oxygen represent current upwelling conditions we see in the Monterey Bay, but also future projected upwelling conditions according to the year 2100, especially prevalent in the extreme treatment. And so we replicated the study again in 2024, but testing the timing and duration of this upwelling exposure and how that affects the partition of the fish. So we have 3 treatments now: ambient, pre-fertilization, and gestational. For this talk, we only need to focus that pre-fertilization and gestational are upwelling exposure, and therefore we would expect the same results for those 2 treatment groups compared to fish that were in the ambient tanks. So for the field study, I used the Coastal Upwelling Transport Index from NOAA, or CUTI, and this measures the total vertical transport of water per day in meters squared. And so here is a graph that I put together of our fishing dates. Specifically, we had 12 fishing trips, and then plotted on the y-axis are those CUTI values. And so a CUTI value that is showing upwelling would be higher than 2.0. But however, this is a constraint of the study. We never actually caught fish during upwelling where it was higher than that 2.0. So that's something to consider with the results. But I took rolling averages of these CUTI values, so 1, 3, and 5, and 10 day. And I selected around 5 rockfish from the fishing trips that matched that upwelling gradient scale. And so looking at our results for 2023, the laboratory study. So here is the graph for HIF1 alpha. So on the x-axis, we have our treatment levels with upwelling stress, and then on the y, we have our protein concentrations within those tissues post-mortem. And so we can see that in 2023, there was a hypoxia response, however, not significant, but we do see that as upwelling stress increases with the tanks and the treatments, so too does the increase in the protein of HIF1 alpha within their tissues. For L-lactate, so for metabolism, we saw no trend among the varying fish. And it's also important to note that we sampled 9 fish per treatment, and those are the same 9 fish that we sampled throughout the experiment in 2023. For cortisol, we saw really no trend. There was a bit of a spike in the extreme treatment, however, not significant. For IGF-1, we saw a very interesting trend. So IGF-1 levels increased as upwelling stress increased, but to a point, and then it significantly dropped off, which supports our hypothesis that IGF-1 levels would be lower in fish that were in the most extreme treatment tank. However, this is not significant, but again, we do see that trend. So looking at our results for 2024, starting off with HIF1 alpha, we do see a significant increase in the concentrations of HIF within the tissue in those gestational and pre-fertilization treatments. So that upwelling stress treatment compared to those with the ambient. So this actually supports our original hypothesis. So for L-lactate and cortisol in the 2024 laboratory study, we saw no differences between the treatment groups. If anything, with L-lactate, we're actually seeing a decrease in those concentrations within the tissue. So for growth hormone, or IGF-1, we saw something very interesting. They actually increased their concentrations within the upwelling stress group, so that gestational and preferred. And this is contrary to our hypothesis. So with upwelling stress, we would expect lower levels of IGF-1. However, we see the opposite. And I think this is because these fish are giving patricians, they're giving birth to live young. They're actually increasing their growth hormone for those larvae. At least we're seeing that trend potentially in 2024. And this was significant. So moving on to our results in the field study. Starting off with HIF-1 alpha. So here is a graph of the QD averages over a 10-day average plotted on the x-axis and HIF concentrations on the y. And we do see a significant decrease in the amount of HIF proteins with fish caught during high upwelling stress, so that high QD value, compared to those caught during relaxation events. But this is actually contrary to our hypothesis. So something else is happening. We're still trying to figure it out. But we would expect HIF to be higher, but it's actually lower in the field. So that's something to keep in mind. And then next we have growth hormone. So this actually supported our hypothesis. We saw decreases in IGF-1 within the tissues of these rockfish when they were caught on high upwelling days. And this was significant. And then I'm still in the process of analyzing the L-lactate and the cortisol concentrations of these fish. So that's, more to come. So what does this all mean? So rockfish are having a physiological response to this upwelling stress. So there's potential acclimation happening, which is good. Yay, they're acclimating to climate change stressors in the lab, in the field. It's cool. But this requires energy that's used for other processes like growth and patrician. So not so great. The susceptibility risk of these rockfish, especially gophers as a benthic species, is probably going to increase due to climate change, potentially increasing the amount of upwelling they experience in the field. But from this project, we can successfully capture how much protein is in the rockfish tissue post-mortem using ELISA assays, which is a fairly new thing to do for rockfish species. And so in summary, HIF-1, going through each indicator, with HIF-1 alpha, we would expect increases in those concentrations with upwelling stress. Overall, we saw that apart from the field study. So there is evidence to support that this could be used as a bioindicator. L-lactate and cortisol, we would expect increases of those as well. However, the results were not really different between the treatments in the lab. And then in the field, we'll see what we get. But there's no evidence to support that these are bioindicators of an upwelling stress response. IGF-1, we would expect decreases, and overall, that's kind of what we saw. We saw it in the field. We saw it a threshold effect in 2023, opposite effect in 2024, but we are seeing differences. So there is evidence to support that they could be used as bioindicators. But yeah, for the field acclimatized fish that we caught for the field study, more work definitely needs to be done, analyzing other variables other than the QD upwelling indices, maybe looking at the dissolved oxygen, the temperature gauges. I still have to sort through that data, but this is very preliminary. And then the next steps for me, for my thesis, is to branch out to pelagic species in the field study. So I want to look at blue rockfish, potentially maybe olive rockfish that aren't so benthic that they could move away from that upwelling stress. And so I expect that those proteins are going to be more even keeled compared to the differences we see in gopher rockfish. So I want to thank all my collaborators, all the special people that made this project happen, and I can take any questions. Thank you.
Awesome, great talk. I love that. Looks like a rockfish matches your jacket in that picture.
Yeah.
Um, no, that was fantastic. Does anybody have any questions for Matthias? Kim.
Hi, great talk. Um, my question is, so on the last graph that you showed, in your results,
Mhm.
it showed yeah, there was like a significant trend there, it said. Um, but looking at the R squared, it's only explaining about 19% of the variation. So I was wondering if you had any like ideas or thoughts on like what else could be explaining variation in the environment and for those IGF-1 concentrations.
Yeah, so IGF-1 concentrations, also, I guess, a reason why we picked IGF-1 is there's um, evidence to support that, um, fish, or specifically rockfish that have higher levels of IGF, so higher levels of that growth hormone, it's directly related to food. And so with recruitment from like barnacles, increases with upwelling, and so we would expect, this kind of higher trend. But in terms of the variation, yeah, I definitely want to look more into the environmental conditions, temperature, dissolved oxygen, pH probes that we measured within the field. Um, and yeah, see those differences. Also, I haven't looked at the lengths and weights yet, and maybe even Fulton's K of those fish we caught in the field. And so we might see trends with, how big the fish were as well.
Thank you.
Mhm.
All right. Tyler, I see that you have your hand up.
Yeah, awesome talk. So yeah, great work. This is super interesting. Um, quick question. I don't know if I caught this or not. How are you capturing these fish in the field? And depending on the particular methodology, how would you say that affects the fish's stress levels and
Yes.
cortisol
Aw-
result?
Awesome. That's a great question. Yeah. So we caught the fish via hook and line. And so we would catch the fish. Um, and then we would, if they were used for the field study, we would on the spot euthanize them. So bonk them on the head. It's the most humane way to do it. And then we would dissect the tissue and then flash freeze it in a cauldron and then transport it back to the lab in the -80 degree, freezer. And then I would perform ELISA assays on the proteins. But all the fish that we caught experienced that same level of stress of, they got caught hook and line, they're transported onto the V board that I have, they're killed, and then within 2 minutes or so, tissue is extracted and then preserved. All of that happened with every single fish. And so we would still see differences in their protein concentrations because they all experience that same level of stress. Yeah.
Great, thank you.
All right. We have time for probably one more question if anybody has any more. I realize the theme for all the students is like sampling in beautiful locations. You guys all did your thesis work correct. All right, if we don't have any more questions, then that wraps up our student symposium. Thank you to all of our students for signing up and participating to speak in this. Thank you to all of our judges for coming. I really appreciate you all. And thank you to everybody coming and watching the talks and seeing all of our presenters. It was wonderful to have you all here. Make sure you get signed up for our webinar series and then keep a lookout for that camping trip I talked about and our poster workshop. Yeah, everyone's talks were amazing. I don't envy the judges. I'm glad I was just timekeeper. So I had an easy job today. Hopefully we will know within the next couple of days and we will let the students know who the winner is and we'll look for the announcement on social media and through email. Oh yeah, there's going to be a tide pool. Thanks, Kim. There's going to be a tide pool event in Santa Cruz, May 18th. The Bridges, what's the name of the place? Sorry. Bridges.
Natural Bridges, I think.
Yeah.
Also, Paige, I'll get back to you. I need to respond to your email, but yes, Paige is the SAC, or not SAC, the
Or
Santa
Santa Cruz
Cruz
content.
subunit is also helping develop that. So,
But yes, if you were judged, please submit your scores to Matt today so that we can get those tallied and let our students know ASAP. Thank you, everybody, and we look forward to seeing you at the rest of our AFS events that we have this year. Have a good day, everyone.
bye.
Thank you. Bye.
Thank you.